Good day, everyone, the organizers and coordinators, all other protocols duly observed. My name is Omokekbele Deborah Sindhia, a research member of Computational and Biophysical Chemistry Laboratory, Department of Pure and Applied Chemistry, Ladoke Akintola University of Technology, Ogbomoso, Nigeria, supervised by Professor M. Abdulhamid. I'm here to present a research work titled "Unveiling the Potential of Aframomum Malegueta Phytochemicals in Lassa Fever Treatment: A Computational Study". Here is my outline. Lassa fever is known to be an acute zoonotic disease of humans, and it is also hemorrhagic in nature. It is zoonotic in the sense that it is transmitted from animals to humans or humans to humans, and also hemorrhagic in the sense that it causes bleeding. Lassa fever is reported to cause about 100,000 to 300,000 infections annually, with about 5,000 death cases. This Lassa fever is endemic in part of West Africa and possesses significant public health challenges due to its high morbidity and mortality rate. Lassa fever is usually caused by Lassa virus, which is actually categorized as an old world arenavirus and characterized by its envelope structure, single-stranded RNA, and bisegmented genome. Lassa virus is actually negative sense and single-stranded RNA. High-risk areas for Lassa fever are found primarily at the western and eastern edges of West Africa, known as the Lassa Belt, which comprises countries such as Guinea, Nigeria, Sierra Leone, and Liberia. Currently now, there are no vaccinations for Lassa fever and no cure yet, but some antiviral drugs have been used for its medication, drugs such as ribavirin and favipiravir. Ribavirin is actually known to be the primary antiviral drug for Lassa fever, but it has some noticeable side effects. It helps in the chain termination and mutagenesis of the viral RNA and depletes the GTP to disrupt the RNA synthesis and capping and modulates immune responses. While favipiravir on its own is still under research, but it helps to target the RNA-dependent RNA polymerase, leading to chain termination and later mutagenesis, which helps to incorporate its active form into viral RNA. These drugs helps to impair the virus activity. To better understand Lassa fever, we would consider the stages of Lassa fever. Lassa fever actually occurs in four different stages. The stage one is known as the incubation period and can last from 6 to 21 days and sometimes 7 to 10 days. At this period or stage, there are no symptoms recorded. This is because the virus is still at its incubation phase in the body. We move over to the second stage, which is known as the early stage, and actually has a duration of 1 to 3 days. At this stage, the symptoms of this fever are gradually developing, and some symptoms such as fever, general weakness, headache, sore throat, muscle pain, chest pain, nausea, vomiting, diarrhea, cough, and some others, which are general symptoms of normal fever, are developed. Then we move over to the third stage, which is at the acute stage, ranging from between 1 to 2 weeks. And here, all the symptoms are developed, symptoms such as high and persistent fever. We also have severe vomiting and diarrhea. We have respiratory symptoms such as cough, chest pain, and also difficulty in breathing. At this third stage, neurological symptoms such as seizures, tremors, and disorientations are also developed, but we have less or less common hemorrhagic symptoms but may include nosebleed, gum bleeding, and blood in the vomit or stool. Other symptoms such as swelling of the face and the neck, conjunctivitis are also shown or seen. At the first stage, which is actually the final and severe stage, it has a varied duration and can progress rapidly. The symptoms that are shown at this stage are usually the multi-organ failure. At this stage, this is where some of the organs have begin to fail, organs such as the liver, the spleen, and the kidney. At this stage, we also have hypertension, which involves low blood pressure. We have shock, severe and potentially fatal shock. Then at this stage, the hemorrhagic symptoms are now fully shown, which involves internal and external bleeding. The neurological complications such as coma or convulsions are also seen at this stage. In order to understand the drug target mechanism of Lassa fever, we would consider the breakdown of the protein nature of Lassa virus. The Lassa virus contains four major protein pathways, which include the glycoprotein, the nuclear protein, the polymerase protein, and the matrix protein. The glycoprotein on its own helps or is involved in the entering of the virus into the host cell, and it comprises of the glycoprotein 1 and glycoprotein 2. Nuclear protein on its own helps to inhibit replication and also is involved in transcription and replication of the virus in the host cell. That is, it encapsulates the RNA genome, forming the RNP complex. The polymerase protein, also known as the help protein, is also involved in replication and transcription. Actually, it helps to replicate and transcript the RNA synthesis. That is, it synthesizes viral RNA in conjunction with the nuclear protein. The last of this pathway, which is the matrix protein, known as the Z protein, coordinates viral assembly and boarding, incorporating GP into the viral envelope. These four pathways are interconnected to each other, and their interconnections are critical for the Lassa virus life cycle, making each of these protein a potential target for antiviral drugs. Now, talking about antiviral drugs, for now, there are no vaccination or drugs that can help to cure Lassa fever, but some antiviral drugs are used for maintaining the symptoms. Drugs such as ribavirin and favipiravir are in use. Ribavirin actually serve as the primary antiviral drug for Lassa fever treatment, which is actually effective if it is given at the early stage, but has noticeable side effect. Favipiravir on its own is under study, and has a broad spectrum of antiviral effective against various RNA viruses, and is also showing the promise of treating Lassa fever, but has not been tested on humans yet. These drugs, with their excellent antiviral nature, but also possesses some side effects. Ribavirin on its own has common side effects that include anemia, fatigue, headache, nausea, and insomnia, with some serious side effects looking at liver toxicity, teratogenic effect, cardiac issues, respiratory issues, and mood changes. Favipiravir on its own has common side effects that includes diarrhea, elevated liver enzymes, and reduced white blood cells count. Serious side effects include teratogenic effect, liver toxicity, and a potential QT prolongation. These side effects are the reasons why there is a need for other therapeutic agents to be discovered. Talking about therapeutic agents, medicinal plants have been researched on to see if phytochemicals or chemical compounds from this plant can be used as therapeutic agents against viral diseases. And it has been shown through research that phytochemicals from various medicinal plants have shown excellent antiviral effect against different kinds of viral disease with little or at times no side effect. Now, the medicinal plant in consideration in this research is actually the Aframomum malegueta plant. And pharmacologically, this plant exhibits anti-inflammatory properties, antioxidant properties, anti-diabetic properties, hepatoprotective, immune-enhancing properties, anti-hemorrhagic properties, and antiviral properties, which are actually needed for the study of Lassa fever. In this research, 250 isolated phytochemicals from Aframomum and Melegueta respectively were tested against two proteins, which include the Lassa virus L protein cap snatching in the nucleus and the Lassa fever virus nuclear protein using computer-aided drug design approach. Now, the computer-aided drug design approach is actually an approach that helps us to design and optimize potential therapeutic compounds. This approach reduces the cost of developing this therapeutic agent and also helps to streamline the processes that are involved in this design and development of this therapeutic agent. And most of all, it has improved efficiency and accuracy. The computer-aided drug design approach is actually of two forms, which include the structure-based drug design and the ligand-based drug design. Our focus for this research is actually the structure-based drug design that involves the use of the structures of both the ligand, phytochemicals, and our target receptor, analyzing their bonding affinities and the strength of their inhibition constant, checking the docking and scoring analysis, looking into their binding site identification, checking their virtual screening, compound selection, lead optimization, and thereby helping us to design a new therapeutic drug. Aim of the research. The aim of this research is to study the isolated phytochemicals from Aframomum malegueta which serve as a probable cure for Lassa fever using computer-aided drug design approach. Materials and method. Under materials and method, the methods that include in this CAD process for this research involves the identification and preparation of our phytochemicals. The 3D structures of our phytochemicals were retrieved and downloaded from PubChem database and visualized or prepared using Biovia Discovery Studio. And 250 of these phytochemicals from Aframomum malegueta were identified and prepared. Identification and preparation of target receptor is the next step. Our target receptor were obtained from protein data bank, and these receptors include the Lassa virus L protein cap snatching endonuclease having a PDB-ID of 5J1N and the Lassa fever virus nuclear protein having a PDB-ID of 3MX2. These receptors were identified, retrieved and downloaded from Protein Data Bank, and the Biovia Discovery Studio helped us to clean the protein by removing some water molecules and some other molecules that may interfere with the analysis. Then from there we move over to the adment profiling and drug likeness analysis. The ligands that are to be worked on with these receptors were analyzed in order to screen out some compounds or ligands that might be toxic. And so the adment analysis was done. Adment involves the absorption, distribution, metabolism, excretion, and toxicity levels of these ligands. Out of these 250 phytochemicals that were retrieved and down isolated at the Hardman profiling analysis stage, only 54 out of these 250 ligands passed the Hardman analysis stage, and Hardman analysis was done using the Hardman SAR2 database. The drug likeness analysis was done using the Swiss Hardman analysis database at Swiss Admin database, and the drug likeness analysis was done putting into consideration the linguistic rule of 5, and this rule states that orally active drugs should not have more than one violation of this following criteria, and that is: one, the hydrogen bond donor of the compound must be less than 5. The hydrogen bond acceptors of this compound must be less than 10. This is because the lesser the hydrogen bond acceptor and donor, the better membrane permeability. Then we also have the molecular mass of this compound should be less than 500. Then the octane water partition coefficient must not be greater than 5. So for our ligand, out of the 54 that passed the Hardman analysis, we saw that using this linguistic rule of 5, only 49 passed the drug likeness analysis. Then we move over to our molecular docking protocol. For the molecular docking protocol, the Pyrex software was used to dock our receptors and our ligands together, and this is to determine the binding affinity in order to get the inhibition constant of these ligands. Then we moved over to the oral availability analysis of the ligands, the bioactivity test, the PAST analysis, then finally the binding mode and molecular interactions of these ligands in order to ascertain the therapeutic ability of these ligands. Results and discussion. The target receptors were retrieved from protein data bank, and Casp and also Discovery Studio were used to analyze the active site of this protein, and the Lassa virus L protein capsnation endonuclease active sites were gotten and shown on the screen, while also the Lassa virus nucleoprotein having a PDB-ID of 3ms2 active sites are also shown. Next, we went over to the Hardman profile analysis. As earlier said, 250 bioactive compounds from Aframomum malegueta were analyzed, and out of these 250, we have 54 of the ligands that passed Hardman analysis. In this Hardman analysis, the absorption and distribution pathways of these ligands were analyzed, the metabolism were analyzed, excretion and toxicity were also analyzed, and in order to determine the Hardman profiling of these ligands, the hepatotoxicity level of these ligands were cross-checked and ensure that these ligands are not hepatotoxic, they are not carcinogenic, they are non-hemostoxic, and also they are biodegradable. These were the criteria that involves in the selection of the ligand, and also on the absorption and distribution aspect, we looked at the water solubility aspect, which is log S, to ensure that the solubility range of our ligands are between -1 to -5. The ligands that passed and the best 10 ligands that passed the Hardman analysis as shown on the screen. After the advent analysis, we went over to do the drug likeness analysis. In this analysis, we considered the linguistic rule of 5, and we ensure that our ligands do not violate more than one of these rules. Now, out of the 54 ligands that passed advent analysis, that 49 of these ligands also passed the drug likeness analysis. And then on this drug likeness analysis, we ensure that their molecular weight are less than 500, their hydrogen bond donor are less than 5, and the hydrogen bond acceptor are less than 10, and they have good octane water partition coefficient, which should not be greater than 5. The ligands that pass this drug likeness analysis and the top 10, including the standard drugs, are shown above. From this analysis, we see that from the advent analysis, we saw that our standard drugs, both the ribavirin and favipiravir, are toxic, hepatotoxic in nature. Now we move over to our molecular docking result. The ligands that passed the advent and drug likeness analysis were subjected to molecular docking using the Pyrex software. Now these ligands were docked against the Lassa virus L protein cap snatching endonuclease having a PDB ID of 5D1N, and the Lassa fever virus nucleoprotein having a PDB ID of 3MX2. For the endonuclease protein 5G1N, we saw that cyclolucalenol, diazine, navigerin, optisulfonyl had better binding affinity than our standard drug. They had binding affinities of -8.1, -7.7 against our standard drugs that had -7.1 and -4.7 respectively. For the Lassa fever virus nucleoprotein with the PDB-ID of 3MX2, we saw that navigerin had a better binding affinity, the most preferred binding affinity of -11.2, while diazine, diosmethyl glycine also had better binding affinity of -10.4, -9.6, -9.0, which are better in comparison with our standard drug ribavirin having a -7.1 and favipiravir having a -5.8. Then we were able to calculate our inhibition constant using the formula Ki equals to exponential minus zeta G over RT, where Ki is the inhibition constant, zeta G is our binding affinity, then R is our gas constant, and T our temperature. After in calculating the inhibition constant, we ensure that the selected ligand had inhibition constant ranging from 0.1 to 1.0 and must not be greater than 10.0. And in this, we saw that our standard drug ribavirin and favipiravir had inhibition constant of 6.3 and 56.3, which is actually going over the range for inhibition constant of 0.1 to 1.0 macromolecule. Then next we carried out our oral bioavailability analysis, and here the ligands that were docked were analysed. We analysed their molecular formula, the molecular weight, the rotable bond, the linguistic violation, the bioavailability score, and the synthetic accessibility. ligands that pass this analysis, and there are top 10 as shown on the screen. Next, we did our bioactivity analysis for each of the ligands in comparison with against the target receptor, and this result is actually shown or displayed in the slide. As part of the analysis in computer-aided drug design, the pass analysis is also one crucial analysis that needs to be done. Pass analysis, which include the prediction of activity spectra for substances, is used in drug design to predict the biological activity spectrum of a compound. And from this analysis, we saw that our compounds, our ligands and selected or 8 compounds have biological activities such as anti-inflammatory activities, they have antiviral activities, they have anti-infective activities, they have hepatoprotectant activities, they have respiratory and eleptic activities, they have the RNA synthesis inhibitor activities, they have anti-hemorrhagic activities, and so many other activities that are vital for the development of this Lassa fever therapeutic drug. Then the binding mode and molecular interactions of these ligands were analysed using Discovery Studios in order to ascertain if these ligands actually bonded with the active site of this target receptor in order to ensure that the inhibition of these receptors are carried out. The ligands and their binding modes and molecular interactions are also shown on the slide. In conclusion, this study evaluated 250 isolated phytochemicals from Aframomum malegueta plants for their inhibitory prowess against Lassa fever receptors. Lassa virus L protein capsid endonuclease and Lassa fever virus nuclear protein using some computational tools such as ADMET-2, Mode Inspiration Web Server, Parrot Docking Tool and Swiss ADMET. The result obtained revealed that cyclohexylamine obtusifolate and dihydroxymethoxyamphetamine from Aframomum malegueta are potential inhibitor of Lassa fever virus L protein capsid endonuclease, while navigerin, diazine and diosmetine are potential inhibitors of Lassa fever virus nuclear protein, all having better binding affinities than the two standard drugs ribavirin and favipiravir. Conclusively, these ligands have preferred ADMET, drug likeness, oral bioavailability and biological activity profile. These ligands are potential inhibitors of Lassa virus L protein capsid endonuclease and Lassa fever virus nuclear protein. Their properties and activities can be further improved during lead optimization in order to make them a probable therapeutic agent for Lassa fever. These are some of the references considered or used. The authors acknowledge all the members of the Computational and Biophysical Chemistry Research Group under the mentorship of Professor Abdulhamid Musbauddin of the Department of Pure and Applied Chemistry, Ladoke Akintola University of Technology, Ogbomoso, Nigeria. Thank you for listening.